Hi, I'm On. I worked in Dr. Virginia Moore's lab this summer, and we specifically looked at cold and freeze-thaw tolerance in a couple USDA pea collections. And we had access to around 400 germplasms, and due to time constraints and also space constraints, we ended up just bulking them into 4 groups and then comparing them to a control, which is called Wild Winter, which is established as a very cold hardy cultivar. And we performed this experiment in a controlled environment, which is called, for us, it was a thermal gradient table, so that's pictured here. And the way we manipulated the temperatures to expose them to cold temperatures were through these valves below. So they basically just circulated very cold solution throughout the table, which changed the soil temperature. And we were specifically looking at 3 things. So damage score, percent of damaged plants, and then also ion leakage. And if you look to the figures over there, you can see that on this top one is our ion leakage, and there's not a lot of variation between our genetic groups, and we didn't find any significant results. In the second table here, we looked at the percent of damaged plants in a plot, and only the control group was significantly different, which was to be expected since it is more cold hardy. And then for our average damage scores, we saw that the control group and the Eladius group were the only significantly different genetic groups, with the control having lower damage and the Eladius group having higher damage. We also did this same thing specifically for plants that were exposed to our lowest temperature, so that was about negative 14 degrees Celsius. To see theoretically they'd be the most stressed out, so maybe they would show something different. We didn't find anything statistically significant with these groups, but across the entire table as seen here, we did. And this is pretty important because in a lot of northern regions, crops are needed to be very cold hardy to survive winter kill and be able to be harvested. And a lot of times peas will not make it through the winter, and some commercially available cultivars are still proving to be inconsistent over time and across different locations. So this is kind of a part of that experiment as well.
Very, very nice work. Thank you. What next steps would you take, or what would be the next experiment you would perform in this work?
Yeah, so as I said, we bulked the group. So the next step would kind of be able to look deeper into the group, so looking at specific germplasms and be able to identify them at a more detailed level. And we'd also like to be able to control or compare what we saw in the thermal gradient table to results in a growth chamber, and then use that to kind of see how far away we are from kind of modeling field experiments, since theoretically we'd want to see how these peas perform in the actual scenario in which we need them to perform.
Great. That sounds like a great future plan. Thank you. Very nice work.